So let's get started reviewing this article, Selective Bird Predation on the Peppered Moth, the Last Experiment of Michael Mejiras. Michael Mejiras is a very famous experimentalist in the area of evolution. And the purpose of this lab, this research article or his research, was to try to set the stage, or at least prove, hopefully, that there was something called evolution or natural selection occurring when the moths that rested in the forest of England and many parts of the world were selected against their white color or their peppered color that matched the lichen color bark of the types of trees they like to rest on during the day. So as industrial revolution started, as we talked about and we saw in our video, the Industrial Revolution had these smokestacks of these factories that burned probably coal, and coal, along with other types of fuels, when there's incomplete combustion, creates soot, and that soot settled on the forest, especially on the trees. By making the background of the trees now darker, it was the now white moths that were selected against. And because the white moths could be easily seen, the idea was that obviously the birds would now eat them more than the darker moths. And of course, the frequency of the darker moths increased. Now, the darker moths, okay, are called melanic carbonia. Carbonia is for the carbon that was given off. Anytime you see dark smoke, it's because of carbon. If you ever had the graphite in your pencil is a dark color. That's also carbon. If you ever had a barbecue with briquettes, not a gas stove, but you actually use the carbon briquettes, those are carbon, those are black, or a carbon-based filter. So that's where they get that name from. So melanic carbonia is the moth that has the black color. And of course, the typica is the white form. Okay, so here typicals and the melanic is the type of moths. But back to what we're talking about here. So they understood, at least they thought they understood, that the dark color that was developing on the trees was allowing for the white moths to be eaten more. They were selected against that background. And so the proliferation of the melanic carbonia increased. Now there was a reversal to this as we cleaned up these factories and we used less of we had less of this type of soot being produced. These trees had less soot on them, and therefore they became their peppered white background bark that we normally had. And that background, that environmental change, selected against the black moth, and subsequently the black moth decreased. And that has been something that's been accepted. Recently, though, and this is recently, the last 25 years, people who have come to understand that have questioned the idea that selection is taking place because of poor experimental designs and poor research in this area. Some people have been able to attack evolution as a whole. As you're probably aware, okay, there are people that believe that evolution has never taken place. So because of the poor scientific work that supported, okay, the decrease in black because of natural selection, the idea of natural selection or evolution as a whole has been actually questioned. So obviously, Michael Mejiras wanted to put that to rest with a comprehensive study that would try to prove, or at least show some information that in fact there was a selective pressure. So he did things a heck of a lot better and was meticulous. Unfortunately, he didn't live to completely publish this. Okay, so that's the background here. Now, was he trying to prove something? And careful, when researchers are trying to prove something, they may in fact, okay, do so without them knowing. That's the bias. But his work was so meticulous, it actually put a nail in the actual wood here, in the idea that there is evolution. So this is groundbreaking work that stopped all the discussion that there was no evolution. Okay, now what did he do? Well, what he did was he used real moths. He didn't pin them on. He also made sure that there were, because there was ideas that bats were taking the black ones or the white ones. And bats use sonar, they don't have sight, so the black or white would not be selected for. And he showed in a separate part of this that the bats took at night took either the white or the dark ones equally. So there was no factor due to the bats. Okay, but there's all kinds of other problems, like people in these other studies never actually watched birds eat these moths. So part of this study, and this is really, really important, was that he first recognized where, in fact, these moths actually, this is my moth, I guess, where these moths actually existed during the day. So he actually climbed trees to see where they actually hung out during the day. Understand that moths tend to fly at night and rest during the day. So he noticed where they rested, and they weren't always on bark. So he climbed trees to recognize where, in fact, they were. And when he did that, okay, again, no one else did that. He came up with these kinds of observations here. These observations led him to believe that, okay, there was no difference between male and females where they like to be, and he found all the nice resting places. It wasn't always on the trunks as reported in earlier studies. So he went out and made sure that he picked very good places for his moths that he released on these different places. He just didn't put them on trunks. Now, how did he go about doing this? Well, he used these things called sleeves. And basically, he released a certain number of moths with different frequencies of white to black. And interesting enough, because the blacks actually are the dominant gene, and the white is actually the recessive, okay, if you see up here. So you think of, wow, there's a lot more white, but actually that's the recessive gene. So there's places, you always think the recessive is going to be smaller in number, probability-wise, yes. But that doesn't mean the frequency is always going to lead to that. Meaning, if you know you're recessive in Punnett squares, okay, I guess you're using big C and little c. So big C would give me the black, and the little c, I can't write in white on white, but the little c would give me the white typica, okay? So interesting enough, there's a heck of a lot more white, even though it's a recessive, okay? So don't always think that the actual number and frequency of the alleles matches the probability, all right? There's a probability of 1/4 in a heterozygous type of cross, okay? So in any case, interesting enough, I know you found that fascinating. Okay, so back to this. So what did he do? Well, now that he knew where the moths like to go, he didn't just focus on putting his moths in trunks or branches or twigs. He used all three places, and he meticulously found places that he saw the actual live moths. These aren't his moths. He actually climbed trees to see where the moths like to be. So basically, in his trees that he picked out that he thought that were that's an ugly tree, but hey, I'm tall. Okay, he put these sleeves around either the twigs, the branches, or the trunks. And in these sleeves, he put his moths in, black or white. And important, he also put them in with the right density, as we talked about. What's density? Density is how much moths in a certain area. A lot of previous study were pumping in a lot more moths into an area than normal. If you increase the density of the moths, you change what happens in nature. Obviously, there's more food. Maybe birds are eating more than normal, and that changes all the dynamics. So he made sure that he matched, okay, the density of the forest with the moths he put in. Now, what he did was he put these moths who were flying around at night in these little screen containers, these sleeves. Now, when he came to take them off in the morning, okay, if they were still flying around or they landed on the screen, he'd remove them. But if they wound up on the part of the tree, then he took the sleeve off. And he counted 4 hours later if they were still there. If they were gone from their resting position, he assumed they had been eaten. But really important in this lab, what he also did, because it's about predation, he had observers, notice the eyeball, okay? He had observers watching to see if certain birds were eating them. Okay, the idea behind the lab was to see if, in fact, the birds, the prey, or the predators, I should say, were selecting against the white or black. So he had people watching with binoculars, okay, to see which ones are eating. Now, he said about 24% of those missing were observed eaten by birds and observers. So observers were sitting there watching, meticulous work, and they counted. So about 24% of the missing moths they accounted for by different birds, and they talked about which birds were left. So that was really, really an important study to see that essentially 24% due to the observers. Okay, really important. So he was trying. Now, what did he do with that 24%? And this is really important. The 24% missing, so to speak, okay, from observers seeing birds eat the moths, he figured, or they figured out which moths they were eating. So not only did they watch birds eat them, they figured out what they ate. Was it the black melonyx or the typica? Okay, or the typicals. Okay, and what they found, there was a lot more of the melonyx, the black colored ones, eaten. And they found the percentage, okay, so to speak, of what the melonyx were eating. And what they did with that is they took that percentage and they noticed basically the melonyx, remember, the birds would eat the white or the black, but if they found the black easier to get, they'd eat them more. It's a selective pressure because now, remember, the Industrial Revolution is over and the bark or the twigs or the branches are not filled with black soot. So you would say that the bird should have a selective pressure over the melonyx, and he was trying to show that. And what he showed basically was that about the melonic, the black colored moths, peppered moths, okay, were 91% survival over the typicas, the white. So there was about, so to speak, about a 10% difference in the survival. So think about that, 91% survival of the melonyx. So there was some, it wasn't the same, all right? So if you think what that means, so let's think what that means for a second, okay? So if you have about, now of course there is some error bars. It's a great little lab here because you're talking about, there's some plus or minus 95% confidence he talks about, but we'll get back to that. But I just want to set this up a little better, okay? If there is about, and of course there's a plus or minus this, but we'll just say 91%. They're saying that the blacks, the melonic colored moths, okay, we'll just put the M here, survived 91% of that of the white colored moths. Now, what does that mean? Well, if we have 100 melonic black colored moths, okay, that means 91 would survive. And that means there's what? There's 9 missing because of selective pressure. Now, if you don't understand what that means, okay, that means of the same number of white moths, 100, okay, so if you had this going on, for every 100 white moths, peppered moths, the typicas, okay, I should put T here for the typicas, or typicals, all right, they would be 9 less, they'd be 91. So for every 100 moths that survived, there'd be 9 less for the melonyx or the black colored moths. And that they determined to make some kind of selective or S coefficient, some mathematical drawing there. So that's what that means. So again, very important that they saw selective pressure in the birds. Yes, there were typicas eaten and melonyx eaten. However, the melonyx survived 91% of that of the typicas, meaning for every 100, okay, there was 9 more melonyx eaten. Doesn't seem like a lot, but that's enough of a selective pressure that if it's maintained, would drive down the frequency of the black moths. And that's essentially what he found. But let's go back to the work here, because essentially what I'm asking you to do is look at the data table. So let's look at this data table, okay, and a lot going on here, and it's pretty complicated, all right? Now, first and foremost, you see the years. You can't really look at the first year because you want to see a change from the first to the second year. Now, they started with a local melonic, that's the dark color, of 0.1 frequency, Okay, that's important. Now, what they saw overall is there was that many typicas and that many melanics. So this is the white color and this is the dark, okay? So this is the dark colored moth. Now, they saw that there was 162 typicals eaten and 31 melanics eaten, okay? Very, very, very simple there. Now, you have to understand though, the frequency of the melanics is far less. Okay, they set that up. And you can do that math by seeing 101 plus 706, okay, and you can see what that percentage is. Now, all right, what happens is the first piece of data, okay, this data right here, this first column represents, and this is important, the data which we would expect if there was no selective pressure. What they did here was they added up the total amount. So 706 plus 101 is 807. That's the total. Okay. Now they found, okay, if there was how many eaten? Well, there's 162 plus 31 eaten. All right. So if you do that math, you would get 162 plus 31 over 807, the total, to be approximately 0.24. That means 24% of the moths were eaten. If you assume there's no selective pressure, then you can take this 24% and times it by what? The number of melanics eaten, right? If that's 101, melanics available. So if you take the black colored moth, there was 101 to begin with. You take 24% of that, so you times that by 0.24, what you're going to get is approximately 24. Now you say, Mr. Grotsky, they got 22, I'll explain. And if you take this 24% or 0.24, however you want to look at it, if I times it by 100, it becomes 24%. And I times that by the typicas that are eaten, or I find 24%, I should do it the other way. If I times the 24% by the typicas available, I should find that 24% of that, which actually comes out to be 169, that's what I expect. So that's what they kind of did. They did that. And you said, Mr. Grotsky, why is it not coming out perfect? Now, look what I'm doing. I'm seeing if these numbers of expected missing equate equally, assuming no selective pressure, right? Yeah. Now, you say, well, why is it not coming out perfect? Now, look what they said here. Frequency of wild melanics obtained in light trap samples. Typical melanic individuals are made available and eaten at experimental site. Yes, expected values under the null and best fit, that's it. And best fit, okay? So what they did is let's make some room here. What they did was they took these values and they made a best fit line to make sense of this. So what they did, and that's the reason why it's not coming out perfect, is they had a best fit line. So yeah, I might have let's say 169. Let's do that black. Okay. And then the next time I do it, I might be lower. And as I do this, I get a what? Kind of a scatter. And what they do is they draw a best fit line between that. So they're just giving you the value at the best fit line. That's why that number is a little lower than I got there. But I want you to understand how they came up with this number. This number right here, the first column, are the numbers you're supposed to get if there's no selective pressure, just by knowing how much is missing overall and taking that percentage, that same percentage against the black colored moths and the white colored moths that are available. There'd be no selective pressure. They should be the same. Okay, so you can see that that wasn't the case. So let's get rid of some of this and let's look at this carefully. So what we have, party people, is we have 154, 159, okay, and you can see that these numbers are a little higher, okay, than others. Now, why would like this one be so much higher? How come there's so less white ones eaten? Well, probably because there's more what? The black ones being eaten probably. Who knows? But the bottom line is, all right, you can see that sometimes these numbers were exactly the same or very close. Now, what you would notice for the melanics is you notice that this number, okay, is a lot lower for the most part than All of these, wow, except for when we start getting to low numbers. So if you look at the data here very carefully, what you expect is a lot lower. Why were there more melanics eaten? Maybe the selective pressure. So although these numbers jump around a little bit, okay, for the typicals, and we call this heterogeneity, okay, these guys are consistently lower, giving us some indication maybe there's more eaten than expected for some other reason. Now the second column, okay, the second column is that column they figured out what should be missing based upon taking the 24% or so observation of deaths by the many types of birds and seeing that the melanics were eaten, let's say, a little more than the white, and taking that data and extrapolating to the rest of the data. I'll explain it again. Remember, 24% of the missing and eaten moths were observed due to people actually watching the birds eat them. And what they did was, from the ratios of the melanics to the typicals eaten, and again, there was more melanics eaten. That's why they survived 91% of that, of the typicals, okay? We talked about that. So they took that data and took that ratio and extrapolated to the other 76% that they didn't see. Okay, it was impossible to see all of them. And these were the numbers, party people, that they expected to get, assuming there was a selective pressure due to the birds. Notice the 162, 162, 204, 200. These numbers are what? Very consistent, very significant. Look at these numbers, 30.32, 31. You compare these values, a whole heck of a level of significance of both, which again, very strong evidence that the selective pressure could be applied and due to the birds because there was an observation. Okay, so that's something to think about and ponder. I'm not here to give you all the answers. I'm here to help you understand what the data suggests. Now, look at this chart here. Fractions surviving. Now, notice the error bars here. Notice how the error bars, and these are actually plus or minus 2 SEMs, okay? That's what they're using here. Standard error, the mean. They have SE right here, but they don't tell you 2. It's definitely 2. Notice the error bars are getting bigger and the overlapping, okay? And notice the smaller error bars. And notice the fraction surviving of the moths jump around, okay? Whereas the fraction surviving of the black moth stayed pretty constant. Why was there a pretty constant selection variable? Was there a constant what? Was there a constant force of selective pressure on them? Now you may say, well, why are these guys jumping around? Well, if you look at, and I'm giving you too much information here, what they found, okay, if you look up the things they talked about a strong evidence of heterogeneity. That means differences, diversity, differences in the non-selective fraction eaten of both moths across years. What that means is year to year, hello, there's a different fraction, okay, of what? The non-selective fraction was eaten, okay, of both moths, okay? So there was a larger number of eaten, okay, but this is important. There was no significant variation in the fraction selectively eaten, okay? So when you think about that, what that means is the birds, or their observation, they saw the predators eat more moths total year to year, but the fraction that was selected against of that total, okay, year to year was the same. Think about that means and how that relates to that graph. Okay, I'm not going to say any more than that. If we look at some other findings, there was strong evidence of overall selection against the melanic form while taking account to year differences in the non-selection fraction eaten. Okay, and again, kind of spells it out. that non-selective fraction is the moths eaten that were just due to, they think, chance. Remember, if we're saying that 91%, okay, of the black moths survived compared to the what? Typicals, the white ones, that means that there's a whole bunch of black moths that are eaten that are not selected for just by chance. They're right there, okay? So we're talking about that fraction. So only 9, okay, out of 100 typicals that live were selected against, so that 91, okay, are not being selected against. So don't think every black moth here was selected against. So there's a fraction of black moths, okay, that are eaten just because of chance. That's important. And that fraction, that non-what, selective fraction, differed year to year based upon what? How much the what? Total amount of moths were missing, or the birds ate that they what? Saw in their little part of the woods. The maximum likelihood selective coefficient, this is what they found for the selective pressure. I just talked about this. And notice I said 91%, but there was a standard error of the mean, 95 confidence intervals that should jump at you. So if I take my 0.091 difference of survival compared to the white or the typicas, plus or minus what? When you plus or minus the standard of the mean, it becomes 0.028 to 157, which means it comes out to be that the survival rate of the black moth is 84 to 97% of that of the white moths, okay? And that is the range of possibilities within this 90% interval. All right, so those are the basics. Still want you to think about what this graph means, especially the error bars, and you have now all the information you need to do to interpret what this means. Think more about this now. Okay, I kind of gave a lot, but I didn't give everything. All right, so good luck with the rest of your interpretation analysis of this beautiful research article. Good luck.